It's a chilly winter night, January 1952. A young woman stands on the shore of Cold Spring Harbor, New York. She lights a cigarette, her third since she's been standing out by the shore. She takes long draws from the cigarette and plays through everything she did over the day, all the tweaks to her experiment that she'll need to make tomorrow as she gets all the data ready for publication. They had almost all the data they needed, and the findings were conclusive. She had pored over them multiple times in the last few weeks, enduring the long, icy silences of meetings with her supervisor, Alfred Hershey. He was never dismissive, but he always seemed to need time to mull over her ideas, reshape them, till he could resuggest them as his own. Nevertheless, they were both pleased by the results of the experiment they'd been running for weeks now. Every time they ran the experiment, the radiolabeled DNA was transferred from the virus into the bacteria, and the radiolabeled protein never was. It was really that simple. She turned it over and over and felt quite satisfied, though not all that excited. Why was she here? Wasting her time and her energy on this cabal of dull men. She'd leave next year, maybe this year, just as soon as she tied things up with this paper. She stamped out the cigarette under her heel and headed back to her car. She had a long day in the lab tomorrow. Best to get some sleep. Welcome to Modified. I'm your host, Dr. Orlando Delange. I worked for 10 years in plant biotechnology research, work that sometimes involved making genetically modified plants. Now I'm a science teacher, training students for future careers in biotechnology and other areas of life science. This podcast is my attempt to share some of what I know and how I think, to guide you through the science of GMOs and hopefully just enough of the history and social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 2, What Genes Are Made Of. If you're tuning in for the first time, I recommend heading back to episode 1 and starting there. Otherwise, let's go. The woman I described in the introduction was Martha Chase, the laboratory scientist who carried out the experiments that definitively showed that DNA, not proteins, is the hereditary material. She worked under the supervision of Alfred Hershey. He was 44 at the time, she was 24, and she was still starting out in her scientific career. They did that work together at Cold Spring Harbor Laboratory in New York in 1952. 90 years and 4,000 miles away from the lab in Tübingen, Germany, where Friedrich Miescher first isolated DNA in 1869, as we learned in episode 1. Alfred Hershey went on to win a Nobel Prize in 1969 for proving that DNA is the hereditary material. Martha Chase was not included in that prize. Rosalind Franklin, who some of you might have heard of, is often cited as a case of Nobel Institute misogyny for her exclusion from the Nobel Prize for discovering the structure of DNA. Unfortunately, Rosalind Franklin had died of cancer before the prize was awarded, and since Nobel Prizes are not awarded posthumously, she would not have been included even if the Nobel Committee had been so inclined. Alfred Hershey was awarded the Nobel Prize in 1969, in no small part due to the experiments that Martha Chase conducted, and unlike Rosalind Franklin, Martha Chase was alive at the time the prize was awarded. My personal hunch is a combination of misogyny and the fact that Martha Chase didn't go on to run her own prestigious research lab. After her work at Cold Spring Harbor, she moved around the country, continuing to pursue genetic research, though nothing as splashy as the experiments she carried out at the very start of her career as a research assistant in the Hershey lab. If you remember back to last episode, the hereditary material is just a name for some substance inside cells that carries information for traits down generations of cells and down generations of individuals, whether humans, seals, or sugarcane. The idea that such a thing exists has a long history, and for the roughly 100 years from 1850 to 1950, most scientists thought that proteins, not DNA, were the hereditary material, with the tide of opinion shifting more and more towards DNA over time, but without solid proof. The trick used by Martha Chase was to make use of the viruses that Alfred Hershey worked on in his lab. These viruses, bacteriophage as they're known, infect bacteria, and since the early 20th century, geneticists had worked with them because it was clear that when they infect a host bacterium, they transmit genetic information that is stored inside the virus particles. Once transferred into the host bacterium, the hereditary material could be tracked down generations, but by this time the word genes was being used, so they would have said that the virus transfers its genes into the host rather than hereditary material, that slightly clunky phrase. This is in fact how most viruses operate. Viruses are essentially mobile packets of genes, which makes them a useful tool for geneticists. And so by the 1940s, it was clear that these viruses, called bacteriophages, infect genes into the host bacteria when they infect them. What wasn't clear is what those genes are actually made of. Remember that all of this is too small to see. Proving what a gene is actually made of would require some careful detective work. Chemical analysis had already shown that a bacteriophage, like most viruses, is just a packet of DNA and protein. So the experiments that Martha Chase carried out have the simple goal of tracking which of these two molecule types is the one that goes into the host bacterium and stays there down generations. Is it the DNA or the protein? The problem is the bacteria have their own DNA and proteins, so how can you tell apart the DNA and proteins that come from the bacterium from the DNA and proteins that come from the virus? All of these events are happening at a scale so tiny that there's no way to discern what's happening by eye or even by microscope. So Martha Chase relied on radioisotopes. Radio for radiation, isotopes meaning a slightly different version of an atom. So a version of an atom that is radioactive. Without going into too much detail on radiochemistry, just know that you can use radiation to label and track specific molecules, in this case DNA or protein, by using versions that contain radioisotopes. You might have heard some of these radioisotopes, things like carbon-14 or phosphorus-32. Essentially, you can grow up a bunch of virus on a bacterial food that is rich in these radioisotopes, and then you will end up with virus particles that contain those radioisotopes after they've incorporated them into their bodies. And we say that these viruses are now radio-labeled. Specifically, Chase and Hershey fed the viruses with phosphate-32 and sulfur-35. DNA contains a lot of phosphate, so phosphate-32 labels DNA. Proteins don't contain phosphate, but they do contain small amounts of sulfur, so the sulfur-35 labels proteins. And then they took these radio-labeled viruses and mixed them with normal non-radio-labeled bacteria, wait some time, and then see which label becomes incorporated into the bacteria after they were infected with the virus. Every time after they grew the bacteria for some days and then isolated the cells and analyzed whether they contained the radio-labeled carbon from the proteins or the radio-labeled phosphate ions that were used to mark the DNA, they found that the signature of radio-labeled phosphate was there, not sulfur. And that showed that it was only DNA that was transferred, not protein. Genes are made of DNA. Definitive proof at last. It was an elegant experiment, and in the immediate years that followed, many other scientists showed in a whole range of ways that yes, indeed DNA is the hereditary material. In fact, just 1 year later, in 1953, James Watson and Francis Crick published a structure of DNA, building on data by Rosalind Franklin. Remember that by the 1950s, scientists were very aware that DNA existed, even if they weren't absolutely sure that it was the hereditary material. And also, solving the impossibly tiny structures of molecules using a technique called X-ray crystallography was not new. It had been used for a decade or so by this point. And so on its face, the structure of DNA was just using an existing method to show the structure of a molecule that people already knew a fair bit about from a chemical viewpoint. But because Hershey and Chase had just shown a year before that DNA is what genes are made of, that 3D structure of DNA, that famous double helix, was an immediate sensation. In 1953, people around the world could visualize and understand what this thing is inside all of our cells, this hereditary material that seems to shape so much of who we are. We all have genes, they're made out of DNA, and its shape is a double helix. A few times so far, I've used the word gene without explaining what I mean by that. And despite my instinct to define every single scientific term I use, I don't feel too worried about using the word gene, because it's a part of everyday language, and it has been for decades. Most people have a sense of a gene as something related to DNA and something that makes you you. Genes are units of biological fate. or, to use that slightly more technical term, units of heredity. At a physical level, what is a gene? That was worked out over the next couple of decades after it was proven that DNA is a hereditary material, and the physical structure of DNA was solved, which immediately suggested a lot of ideas about how it might function at the molecular level. For instance, DNA is a double helix, meaning it forms a structure of two strands or chains, each with an identical repetitive structure of sugar phosphate backbone units that directly links together. And then there is a bit sticking off the side of every unit, and we call that the side chain. For DNA, those side chains can be A, G, C, or T. Now, those are just letters that we use as a shorthand for four similar but slightly different carbon-based molecules, and they have the longer names adenine, guanine, cytosine, and thymine. And while I know this may seem obvious that our cells are not full of little letters, years as a science teacher has taught me not to shame or belittle anyone for a misunderstanding. Because whenever DNA comes up in visual media, the image is normally either a double helix or it's some matrix of letters, those A's, G's, C's, and T's. And indeed, scientists who work within the world of DNA do tend to think in terms of sequences of letters and don't concern themselves too much with what the physical reality is that those letters represent. Those letters are referred to by scientists as DNA bases. And yes, there's a logic to where that word comes from, but it's not really worth spending time on. So you can just know that DNA has these four different bases, the side chains, A, G, C, and T. A base plus the constant sugar phosphate section is called a nucleotide, technically, but to avoid having to say the word nucleotide too often, we'll just use the word base to mean one unit of DNA that's either an A, G, C, or T. But back to our DNA molecule. You have these two chains made of constant identical sugar phosphate backbones with different side chains. The side chains point at each other in the middle of the twisting helix, and that's where the real molecular magic happens. The bases are attracted to each other and form strong molecular bonds, but only in two specific combinations. The two combinations that form complementary pairs are A to T and C to G. That complementary base pairing, that molecular rule that A pairs with T and C pairs with G, was far more exciting than just showing that DNA is a double helix. A double helix is a nice shape, but complementary base pairing, or as it's sometimes called, Watson-Crick base pairing, is fundamental to how DNA works, and as we'll see, how genes work too. Imagine you took a DNA molecule and separated its two chains. Now you have just one chain of sugar phosphate links, and all these side chains just hanging out in space. A's, G's, C's, and T's, in a seemingly random order up the chain. That is unstable. DNA that is naked like that, or single-stranded as we call it, is quickly dissolved inside a cell. But because of complementary base pairing, it would be very easy to fix this problem. You'd know how to form the matching DNA chain by just going along one DNA base at a time. Wherever you find an A, put a T opposite it. Wherever you find a G, put a C opposite it. T opposite A and C opposite G, and then just stitch together all the sugar phosphate backbones, and that is actually how DNA gets replicated in all cells. The two chains are pulled apart, and then complementary base pairing allows new complementary chains to fill in the gaps, turning one original DNA molecule into two, each one identical to the original, each one containing one chain from the parent molecule, and then one newly synthesized through the molecular logic of complementary base pairing. And the reason that this is so important to genes is that it allows DNA molecules to be copied indefinitely between cells in one body and down generations, all while preserving the exact same sequence of A's, G's, C's, and T's in the original. And in that process, genes are protected, preserved, and passed on. So now, reconnecting all this talk of DNA bases back to genes. What is a gene? A gene is a section of a DNA molecule, a series of anything from a few hundreds to tens of thousands of DNA bases. So now let's reconnect all of this background on DNA bases back to genes. A gene is a section of a DNA molecule, a series of anything from a few hundred to tens of thousands of DNA bases. And they have a specific sequence, with that sequence defining the functional impact of the gene. So picture it. You may know that humans have 23 different chromosomes, with 2 copies of each in most of our cells. Each one of these 23 chromosomes is a separate DNA chain, a double helix, and each one is roughly 100 million DNA bases long. Within those 100 million bases and one chromosome, there are a lot of genes. And there's also a lot of DNA that isn't genes. If you started at position 0, the start of a chromosome, arbitrarily defined, and you start going up the chain, you might find a gene starting at position 50 that goes until 750, so 700 base pairs, and then another one that starts at 1200 and goes till 2500. And intervening between these genes is sections of DNA that is not a gene, can have other functions that, could spend a whole podcast going into all the other stuff that DNA can do apart from being a gene. This process of kind of scanning up a chromosome and seeing where the genes are is something you could actually do right now with a little bit of training and a computer, because thanks to decades of research and billions of dollars of government research funding, there are publicly available maps of the human genome, the genomes of many, many organisms on our planet. But let's say somehow you could actually look inside a cell, and you weren't working at a computer. You're looking into the cell, and you did that same thing of looking along a DNA chain. You wouldn't exactly see signposts that says gene starts here or gene ends here, but there would be some visible signs. Specifically, you'd see large molecules called transcription factors congregating at the start of the gene, in a section we call the promoter. We'll learn a little bit more about transcription factors in episode 4. Then in the middle of the gene, you would see that the DNA double helix has been pulled apart, and a large molecular complex, we call this RNA polymerase, would be chugging away through the central section of the gene. Trailing behind would be a newly formed strand of something that looks very similar to DNA, but has a few tweaks to it, and that would be a molecule of RNA. RNA also has bases that are almost the same as DNA, and crucially, RNA molecules can be made from DNA templates using the exact same complementary base pairing that allows DNA replication to work. So we can say that the RNA molecule that is being made at this gene is complementary to the DNA sequence of the gene. Specifically, the central section of the gene, the bit we call the coding sequence, and it's called that precisely because it is the sequence of DNA that contains the information or code used to make the complementary RNA molecule. Then at the end of the gene, you'd see all of this complex molecular ballet ceasing, molecules disassociating from the DNA, and the two DNA chains being zipped back together into a nice double helix. That end section is called the terminator. The promoter we saw earlier contains specific sequences of DNA that the molecular machinery of the cell can read to know where to start the process of making an RNA molecule, and the terminator has different sequences that give the stop signal. So the promoter and terminator basically frame and define the section of DNA that is used to make a complementary RNA molecule. And that information is retained down generations because the sequence of DNA is preserved as it is copied. So there we have it. A gene is a section of DNA that has all the information necessary to make an RNA molecule that will have a specific sequence of RNA bases complementary to the DNA template. Okay, so what do we do with this RNA? Well, RNA is very cool and deserving of its own podcast. For now, we'll just skip right over it and explaining why this matters, or at least the most important reason. Because ultimately, the RNA is short-lived and itself is just used as a template to make a totally different type of molecule, a protein. So while DNA and RNA are very similar at the molecular level, RNA and proteins are not. I could spend another 20 minutes or so explaining exactly how this works, but for now just know that the sequence of bases in the RNA is used as a guide for a different set of molecular machinery to make a specific protein molecule, reading and translating the information in the RNA into the molecular language of proteins. And indeed, translation is the scientific term used for this process. Going from DNA to protein is called gene expression. If a gene is expressed, that means that inside the cells of that organism, the DNA of that gene is being constantly used as a template to churn out complementary RNA molecules that are then translated to corresponding protein molecules. Proteins are similar to DNA, and they're also long chains made of similar subunits, as in similar to each other within the chain. But whereas DNA is chains of A, G, C, or T, protein chains include 20 or more different types of subunits. These subunits are called amino acids, They are also more diverse than the 4 DNA bases, both in terms of physical shape and size, and also chemistry. So even though proteins are all just chains of amino acids, they are functionally extremely diverse, and they do most of the actual stuff involved in being alive. In episode 4, we'll see some specific examples of how these genes work, and we'll recap on how genes make proteins and how those specific proteins contribute to making a plant. And then later on in the series, we'll look at examples of specific genes used to modify crop plants and the proteins that those genes encode. So, if you're wondering why you've been learning about basic molecular biology when you thought you would be learning about GMOs, it's because I want to make sure that when I start diving into talking about EPSP synthase, a protein produced by GMO corn because of a transgene and how that makes them herbicide tolerant, I'll be able to go into the molecular details of all that without completely losing anyone. So anyway, that is the physical side of genes. A gene is a piece of DNA that codes for RNA that codes for a specific protein. Or more simply, a gene is a piece of DNA that codes for a specific protein. And proteins are molecular machines that shape the traits of living things. In school, you might have learned it as a gene codes for a trait. And just to recap, traits are the things we can see that define an organism. E.g., you could say a gene for eye color or a gene for grain size. That is kind of a relic of how genes were first studied in the early 20th century. There was no other option but to connect each gene to a single trait to make it possible to conceptualize and track the effect of the gene. However, to try and summarize decades and decades of research in 3 words, biology is complicated. Almost every trait, especially at the level of traits that we humans could see or care about, is influenced by multiple genes. Each trait is the product of many proteins working together, influenced by each other and by the environment. That's because the information genes carry is not for a whole trait, but just for one protein. But in cases where things are kept identical, except for tweaking just one gene at a time, then you can often see that effect in the observable traits of the organism. That is never really the case in nature, and in real human populations, but it is the case in selective breeding, where humans deliberately inbreed populations of plants and animals till they are genetically identical, so then when a change, or we call it a mutation, arises in one gene, you can actually see that impact as, say, a change in flower color. So as you'll see in the next episode, episode 3, if you happen to be a patient Augustinian friar named Gregor Mendel, you can track and study that flower color variant and deduce a set of rules about the inheritance of things you'll at first call factors and what will later come to be known as genes. The machinery that supports gene replication and gene expression is remarkably similar across all of life, which is one of the reasons it's almost certain that all living things on Earth are directly related to one another, in a literal sense. Every bacterium, human, and mushroom on Earth are family. Distant family, sure. You'd have to go back billions of years to find our common ancestor, but family just the same. We all have genes made of DNA, and we all have a pretty similar set of machinery to read the DNA to make proteins. And that's DNA's primary job, because otherwise it can't really do anything. The proteins are the exciting stuff. The 20th century was an extremely busy period for biology. In the 19th century, biology was a newcomer to the natural sciences, struggling to compete in prestige and attention against the more well-established fields of physics and chemistry, which had already more than proven their own value to human life and to making money. The industrial revolutions of the 19th century were intimately tied up with scientific research into physics and chemistry, and both supported in turn by mathematics. Biology had contributed relatively little of value. The study of living things didn't seem to be yielding much, even to the field of medicine, the most obvious area of application. That shifted fundamentally over the 20th century, with DNA at the vanguard of that change. In our next episode, we'll explore Mendel's pioneering work in what we now call genetics, but that work was largely ignored at the time and underappreciated until its rediscovery at the very start of the 20th century. In practice, genetics as a field of biological research was born in the first decade of the 20th century. In the year 1900, a trio of plant scientists read and promoted the work of Mendel, triggering broader scientific interest in the topic. In the year 1900, a trio of plant scientists read and promoted the work of Mendel, triggering broader scientific interest in the topic. And in the year 2000, a century later, the first working draft was published of the human genome, with the Human Genome Project wrapping up 3 years later in 2003 with a well-annotated, publicly available digital database of a complete human genome. The result of almost $4 billion of government investment, as well as a private corporate interest, and the work of thousands of scientists across multiple countries. And all the way across that century, from the birth of genetics to the Human Genome Project, incremental discoveries were made, the work of thousands and thousands of people, some of them incredibly famous, but most of them anonymous to all but colleagues in their particular niche field. More and more over the century, DNA moved into the center of biology. There are many biologists who spend a whole career interacting and thinking about DNA only rarely if ever, but they are numerically in the minority. Most biology today is molecular biology, both the basic research that mostly happens at universities, as well as the applied research that happens in the R&D labs of private companies. Molecular biology isn't a field of study, but a category of practice. It is biology that takes place in laboratories where questions are answered and problems are solved using biomolecules, most notably DNA. More and more, this work in the laboratory is connected with work at a computer, studying and designing gene sequences, or the proteins encoded by those genes. One of the common methods used by molecular biologists is the creation of genetically modified organisms, though that usually means genetically modified bacteria or other single-celled organisms. One of the most effective ways to understand how a gene functions is to deliberately modify it and see what traits inside the cell are impacted by the modification. That is one of the reasons that many biologists can get frustrated and defensive about opposition to GMO crops. If genetically modifying organisms is part of your everyday work, then it seems crazy to say that GMOs are something dangerous and unknown. And it can seem more frustrating to those biologists who are working directly or indirectly on saving lives through improving understanding of the human body or developing new types of life-saving treatments for everything from cancer to COVID. Genetics and genetic modification have embedded themselves into the heart of biology. Thank you for listening to Modified. This was episode 2, What Genes Are Made Of. It was written by me, Orlando DeLange. I used many sources to help me research this episode. I want to particularly acknowledge the Cold Spring Harbor Laboratory Oral History Collection, which I used for inspiration in the section of Martha Chase and Alfred Hershey. This is a tiny one-person podcast, so please leave a review if you're enjoying it, and tell a friend. If you have notes, well, I'd love to hear them. You can email me at orlando@modifiedpod.com. That's orlando@modifiedpod.com. I'd love to hear from you. You can find out more about me and the podcast on our website, modifiedpod.com. That's modified, P-O-D, all one word, .com. That's the end of today's episode. I'll round things out with 5 take-home messages, so if you're stopping here, thanks for listening. Until next time. For those sticking around, here are your 5 take-home messages. Number 1. DNA is the hereditary material, and this was finally proven in the 1950s. Number 2. Physically, DNA is 2 long chains that form a double helix. The chains are connected by a complementary base pairing, A's to T's and G's to C's. Number 3. Complementary base pairing allows DNA to be replicated indefinitely while maintaining the same sequence of A's, G's, C's, and T's. Number 4. Complementary base pairing also works to create complementary RNA molecules. This is how genes work. They are sections of DNA that code for a specific protein via an intermediate RNA molecule. Number 5. Proteins are molecular machines that play a dominant role in defining the traits of an organism, so it is a reasonable shorthand to say that genes define traits. Okay, that wraps up our second episode. I hope you'll listen in for episode 3, the one with Mendel and his peas. Bye!